Hello everyone, myself Deeksha Singh Rajput. I am student of JNKVV College of Agriculture Rewa. Today I am presenting a topic hybrid seed production of cotton under the guidance of Dr. R.P. Joshi sir. Here is the general introduction of cotton. English name cotton, vernacular name kapas, scientific name Gossypium herbaceum. Cotton belongs to family of Malvaceae. Cotton is a soft, fluffy, staple fiber that is grown in a ball or protected capsule around the seed of cotton plants. The fiber is almost pure cellulose under natural conditions. Preface. India is the pioneer country for production of hybrid cotton. The first commercial hybrid cotton, Hybrid 4 H4, was released in 1970 from Main Cotton Research Center of Gujarat Agriculture University. Later on, several hybrids were released for cultivation in different agroclimatic zones of the country. By now, 60 hybrids have been released by public sector. In addition, BT cotton hybrids have been developed and released for cultivation by various seeds. Cotton is a major fiber crop for global significance. It is grown in tropical and subtropical regions of more than 80 countries. The major cotton producing countries are China, India, USA, Pakistan, Uzbekistan, Turkey, Brazil, Greece, Argentina, Australia, and Egypt, contributing about 85% of global production. India has the largest area under cotton cultivation in the world. Types of hybrid produced in cotton. First is conventional type. Majority of hybrid released so far are conventional ones. Development of such hybrid involve three steps, that is identification and growing of male and female parents, then emasculation of female parent and pollination of female parent with identified male parent. Cotton is often cross-pollinated crop. The average outcrossing is 6%. The pollen is heavy and sticky and hence cross-pollination occurs only by insects, that are honeybees and bumblebees. Male sterility based type. The development of hybrid using male sterility eliminates the process of emasculation since the anther are sterile in female parent without pollen. Thus, the cost of hybrid seed production can be reduced. In cotton, mainly two types of male sterility such as genetic male sterility and cytoplasmic genetic male sterility are used for seed production. Then comes agronomy in hybrid seed production. First is site selection, in which soil should be medium to heavy, deep, well drained is considered ideal for seed production. Isolation distance. The cotton hybrid seed production plot should be 30-meter isolation distance on all sides from other field. The isolation distance between parents should be minimum 5 meter. Fertilizer. Recommended dose of fertilizer for cotton is 60 is to 30 is to 30 NPK per hectare. Raising of parental lines. If male sterile lines are used as parents for producing hybrid seeds, following eligibility requirements are to be considered. First, an inbred line to be eligible for certification shall be from a source such that its identity may be assured and approved by the certification agency. Second, hybrid seeds to be certified should be the progeny of two approved inbred lines, one of which shall be male sterile. Sowing. Breeder or foundation seeds of selected hybrids, female and male parents are planted in the same field in separate lots with 5-meter isolation distance between parents and 30-meter from other cotton crops. The sowing dates of parental lines are also adjusted in such a way that there is synchronization of flowering in female and male parent, and there will be continuous supply of pollen till the crossing season is over. Steps in hybrid seed production. First, emasculation of female parent. This is a highly skilled, labor specific, and complicated process. Different emasculation methods have been devised which can be adopted depending upon flower types. Flower buds which are likely to open the next day are chosen for emasculation, for which the best time is after 1 pm. This is pictorial representation in which first picture shows bud selection, second shows removal of corolla and anther sheath, third shows emasculated flower bud, and fourth shows covering it with red bag. Crossing of parents. Crossing work is to be started 1 week after flower initiation. The emasculated bud are covered with red colored tissue bag for easy identification. The emasculated buds are pollinated the next day between 8 to 11 am because stigma receptivity is maximum during this period. Nevertheless, it could be extended till 1 pm depending on stigma receptivity. The crossed flower are again covered with a white colored tissue bag to distinguish them from emasculated bud awaiting cross pollination. A thread is tied to the pedicel for identification of crossed ball at the time of picking. Fertilization occur after 12 to 30 hours of pollination. Hence, the cross bud should remain covered for 3 to 4 days after pollination. Again, this is pictorial representation of pollination of female parents of producing hybrid seeds. First is dusting of pollen, then pollen dusted. Third is covering it with white bag, and fourth is ball setting. There are some points that should be kept in mind. If first is if proper isolation is given and early morning removal of open flower in female parents is assured. Bagging before and after is not required. Second, all these operations such as emasculation and pollination are to be undertaken by properly trained person and technical staff. For good setting and ball development, it is necessary that crossing program should be confined to about 10 to 14 weeks after commencement of flowering. Light and frequent irrigation should be given during crossing and ball development. Picking of balls. Only the completely opened balls should be picked, collected in bags, properly cleaned, and subsequently stored in gunny bags after proper tagging. Damaged or undeveloped balls, locks should be separated and removed before sorting. The good balls fit for obtaining seeds should be sent for ginning and subsequent seed certification. Thank you.